Okay. See here. Okay. Hi everybody. Can everybody hear me okay? Yes, yes, yes. Everyone says yes, yes, great. Okay. Good. Let's see here. Let's fix this. And let's share my screen. I know you guys are probably really excited. Break, spring break starts next week. So I'm sure you guys are all looking forward to having a bit of a break, and hope you enjoyed having a bit of a break last last week too, given lecture on Thursday was a documentary, and then Friday lab was canceled because of illness. So that's nice, to kind of like a little mini spring break before spring break happens, right? So I think I'm sharing this screen. Let's make it big. Yep. And then let's make this smaller here in the corner. Okay. So I have not graded the lecture, or sorry, I have not graded the documentary assignments yet that were due on Friday, but I should be doing that probably by the end of this week, that those grades should be in. I did finish grading the first lecture exam though. And I'm happy to report that many people did super great on that lecture exam. So keep it up. So yeah, I had 14 A's, 6 B's, 9 C's, 4 D's, and 5 F's. So because of this, I'm not going to go through each question on the lecture exam today, since most people, well, first of all, the answers are there for you. So if you pull up your attempt, you'll be able to see how you did on the exam and what questions you got wrong, things like that. I will say though, that if you got an F, so anybody who got a C, D, or an F, I would like to meet with you to go over your lecture exam. So I have office hours this Thursday at noon. So what I'm asking you to do is, if you did receive one of those grades, to please send me an email so I can set up a time to go over your exam. And if need be, if enough people email me and we have to go longer than the normal office hours, that's fine too. But yeah, that I think would be the best way for me to help you individually who didn't do great on the exam, to kind of figure out why. One question that I was surprised that a lot of people got wrong, is there was a question about, okay, there was like a word bank with definitions, and like some of the words were sporophyte plant or gametophyte plant, and you had to match what the definition was with the correct terms. And so many people got wrong the responses for spore and zygote. So lots of people wrote that the spore, or chose that the spore is a cell that grows into a gametophyte plant, or sorry, a cell that grows into a sporophyte plant. And a lot of people wrote that a zygote is a cell that grows into a gametophyte plant. That's not right. That's not right. I couldn't believe how many people got that question wrong. So a spore is, what does a sporophyte plant make? What does a sporophyte plant make? And what does a gametophyte plant make, right? So sporophyte plant is a phyte, or a plant that makes spores. Plant that makes spores. Gametophyte plant, or a phyte that has gametes on it, is a plant that makes egg and sperm that can then fuse to become the next sporophyte generation, right? So if you got those questions wrong, I suggest you go back to, I think, the second lecture where we talk about alternation of generations, because understanding those components of alternation of generations is a big part of this class. So if you're unsure now what a spore is and what a zygote is, it's going to really not do you well going forward. So you should really go back to that second lecture and figure that out. But most people did well, and I was very happy to see that. So for the most part, keep up the good work. And for those who didn't do so hot, please email me so we could set up a time for us to meet individually so we can go over your exams. Okay. This is just to show you what I have said before in class. So often, whatever average grade you're getting on your assignment tends to really indicate how you're going to do well, how you're going to do on the exam. So, if you're averaging, 80s and 90s on the assignment grades, chances are, so here on the x-axis, you have your average assignment grade, and on the y-axis, you have the first exams grade. So you could see there's pretty strong correlation between how you do on assignments and how you do on exams. So if you're not doing well on the assignments, chances are you're probably not going to do so great on the exams either. So it's another thing to keep in mind in terms of how much time you need to spend with the material to prepare for exams. Okay. Just as a reminder, this is how your grade is calculated in this course. This is from the syllabus. So each exam is worth 15% of your grade, and there's 2 lecture exams and then a lecture final. And there is 2 lab exams, the first of which is now going to be held after spring break. A question in the chat box, or I thought I had one. First lab exam is this Friday. Oh, no, yeah, it was. So the first lab exam was originally going to be held this Friday, but now because we had to miss lab last week because of illness for the TA, this coming Friday, you guys will cover the ferns, and then the first lab exam will be after spring break. Okay, so this is a good time to move forward from this. So we still got a lot of course left. So if you haven't done so great so far, don't be upset. There's still plenty of time and plenty of opportunities to get more points. For example, the phenology assignment. So the phenology assignment is something I do every spring, and it's we'll talk about it a little bit here. So this is an assignment that you're going to do over the course of weeks. And it might sound like, oh gosh, it sounds like so much work. It's not so much work. It's just, it takes, plants don't change all that fast. So you have to kind of take this over weeks. So it technically starts today, meaning the observation period starts today. And it's going to be due at the end of April, the last day of class, So let me tell you what it is. Right. Well, first of all, the purpose of this assignment, the most important reason we do this assignment is because I want you guys to get outside. Spring is a really beautiful time in our area, and a lot of things change really, really quickly and respond to spring because we have longer day length and higher temperatures. And so plants are going to start waking up and going through all these changes over a really, really short period of time, and often producing some very beautiful flowers. And for those of you who didn't do so great on this last exam, you'll be happy to know that this assignment, the phenology assignment, is worth the total of 2 in-class assignments. So each in-class assignment is worth about, I don't know, just over 2% of your course grade, but the phenology assignment will be worth 5% of your course grade. So that's the same amount that attendance is worth. So that means between this phenology assignment and attendance alone, that's 10% of your course grade. That's a whole letter grade. So keep that in mind. All right. So what is phenology? Phenology is a study of how things change over time. So for example, this is what most trees that are outside right now look like, right? No leaves, just kind of sticks, right? Trunk and branches, not too exciting. However, when spring happens, the plants that have lost their leaves in the winter are going to grow not only their leaves, but also sometimes flowers and things like that. And so you don't go just from, having sticks to being fully covered in leaves, right? It's not like one day, there you go. It does take some time. And so that's what I want you guys to document. Yes. So this is the definition of phenology. It's the study of cyclic and seasonal natural phenomena, especially in relation to climate and plant, what did that say, and animal? Yeah, and animal life, right? So this is a good example, little cartoon on the bottom, kind of showing you what type of changes a plant might experience, for example. Right, so here's somebody out in the field like, oh, let me document what's happening right here. All right, so here's what you guys are gonna do. You're gonna select 10 trees, or 10 shrubs is also fine, but 10 plants that you encounter every day or walk by every day. These can be 10 plants that are, if you happen to have a yard, this could be 10 plants in your yard. This could be 10 plants just right by your apartment, right? If you're living on a block, right? These could be 10 plants on campus. Granted, probably most of you aren't going to campus right now, so that might not be a good option. But maybe you're going to work and there could be 10 plants that you pass every time that you go from where you are to your work, right? They could be anywhere. The point is, pick 10 plants that you're going to walk by anytime you're going to these things, right? Maybe there's 10 plants that you pass when you go from your house to the grocery store, right? Maybe when you park in the grocery store parking lot, if you're out in that part of Jersey, there's 10 plants in the parking lot, right? So pick plants you experience and you don't have to go find, right? And like I said, it's okay to use smaller trees. You could use smaller trees. You could also use deciduous shrubs, right? Shrubs that have lost their leaves for the winter and are going to start to wake up. Okay. On the first day, right? So like sometime this week, you should take photos of all 10 of your trees. And the first day, you're going to take a picture of the tree. And I'll show you what habit and bark mean on the next slides. And a picture of you with the tree, right? So on day one, which could be any time this week, a picture of you standing next to each tree, selfies are fine, and a picture of the tree habit and bark, which I'll show you on the next page. So that'll be like your observation one, day one. Here's what it might look like. So here's an example of what your day one trees would look like, right? So like on the first here I'll annotate. Right? Here is tree number 1, right? Here is tree number 2. You guys will do a total of 10 trees. Whoops, wasn't a very good line. 10 trees total, right? And this is day 1 photos. So here is a picture showing your tree's habit, meaning its form. This is why it's good to use smaller trees if possible, but larger trees are fine too. Here's a picture of you, right, next to your tree. And here's a picture of the tree's bark. So those are what you're going to get on day 1. So day 1 is a total of 2 pictures total. 2 pictures total.
So we should find someone to take our picture?
Selfies are fine. You can totally take selfies. Just try to make sure that, if you're going to, it's great to give me a selfie where you're looking cute and all, but make sure I can see the plant as well. So if you're doing a selfie, you may not be able to see the whole plant. Maybe you want to take a selfie of you like, touching the bark. And so that could be it. Look, if you're going to do a selfie, if you don't have somebody take your photo, if you're going to do a selfie, maybe do a selfie of you with the bark, right? And then another picture showing the whole tree without you in it. But selfies are totally fine. There's a question in the chat box. They have to be 10 different species, right? No, they don't have to be 10 different species. They could be 10 of the exact same species. It just has to be 10 different individual plants. So if all of them happen to be the same species, that's fine. Doesn't matter. If half of them happen to be the same species, that's fine. It's actually a little difficult right now to tell what species you're looking at since there aren't any leaves on most of these trees. So it doesn't matter if they're all the same species. Okay, so you're going to do this. So the first day you're going to do what I just showed you, right? This is the first day, and it could be any day this week. It doesn't have to be today. And then once a week from now until the end of class, you're going to take a picture of your trees. You don't have to be in it. You don't have to be in the picture every time you take a picture every week after, but just once a week, take a picture of your 10 trees. and especially look for changes that are occurring, right? So you're going to look for any new things that weren't there last time. And then after that's done, when that's done, you're going to use, well, you should use an Excel spreadsheet to kind of keep track of your 10 trees. So you'll have trees 1 through 10 and the dates that you take the photos and anything that changed. Sometimes you might say, like, let's say you took a picture of a tree today, right? And then a week from today, you took a picture of it again. And let's say that nothing changed in that week. That's fine. You could just say no changes, no changes. But if leaves show up, you can write leaves. Here's some things that you might see over the course of these next few weeks, right? So you might see buds appear, right? Sometimes there'll be leaf buds. Sometimes there'll always be flower buds, but sometimes you might see leaf-like things coming out of them too, right? For example, here, this green stuff kind of looks like leaves coming out. This fuzzy leaf here, this is a bud. Underneath that is going to be a flower that pokes out. So you might see buds. You might see, and hopefully you see flowers. I really hope that some of you guys end up picking trees that give you a nice, brilliant display at the end of the semester, because it's very exciting when that happens. But most of the trees that you see will at least produce leaves, unless they happen to be dead. So that's another thing. If there's a chance, because you're picking trees right now that have no leaves on them, there is a chance that you might accidentally pick a dead tree. If that happens, it's no big deal. If you think that's happening, you can send me an email. We could figure out what we should do. But leaves is most likely what you'll end up seeing for all of your trees. So the observation period starts today, so this week, and ends 2 weeks before the end of the semester, right? So it ends on April 15th. That way you have 2 weeks after you're taking photos. That gives you 2 whole weeks to compile your submission for me. And so you're going to put all your photos for each tree into a PowerPoint presentation, and you're going to save that presentation as a PDF so that it's less space. And that along with the Excel file are what you're going to send me on the 29th. So there is this is just an introduction to this assignment. There is a full description of this assignment on Blackboard. It's already available, or it will be available, I think, today at 11:30. Yeah. So read over that assignment. And if you have any additional questions, you feel free to email me, or we could talk about it on Thursday. In the full description on Blackboard, I also give you an example of the Excel spreadsheet, like what that should look like. Another thing is, please remember to back up your photos, because if like you're taking the photos and then you lose your phone or something, and then I don't have the photos, I still can't really grade that. So make sure you back up your photos regularly. I'd recommend uploading them to like Google Drive or something like that, just so they're there. Okay. So that's phenology assignment.
We only need a selfie with the tree during the first photo.
Correct. You only need a photo of yourself with the trees the first time. If you want to include yourself in future times, that's fine, but it's only the first time.
Thank you.
Yep. Okay. So that's the first. We covered how people did on the first exam. We talked about the phenology assignment. And now we're gonna go on to the next part of the course, which is the seed plants. We're finally, I can't even believe it, in the seed plants. I don't know if we're gonna finish all these slides today, but I'm gonna do my best to get through at least some of it. But some of it is a little, we're gonna be covering things that are new today, because we have never talked about seed plants yet, and we've never talked about ovules yet. So some of this is a little more dense than some of the things we've covered so far. So we'll see how we're going along, and then I might just decide to finish it on Thursday. Right. Yes. So, oh boy, look how far we've come. Look how far we've come so far. Non-vascular plants done. Spore plants done. Wow, finally in the seed plants, right? Which are still vascular plants, right? They're still vascular plants. But all the rest of the plants we talk about for the rest of this course are going to be in the seed plants. Oops. Okay. Oh, yeah, I do that sometimes. Okay, yeah, we're in the seed plants. Oh, and so in the seed plants, there's two types. There's two types of seed plants. There are the gymnosperms, which are the type we'll talk about first, not this class, but in next classes. And then the angiosperms, which we'll talk about as we finish up the semester. So we did in the first class, the very first class, we talked a little bit about how plants are important, right? And in particular, how they're important to humans. So not only is seed plants an evolutionary milestone for the plants in general, but the seed plants are super important to us as well, right? Because the majority of the things that we eat that come from plants are from the seed, they're either the fruit that carries the seed or the seed itself. The seed plants, as we also mentioned in the first class, are most, so I should say, most of the plants on Earth are seed plants. And furthermore, most of the plants on Earth are flowering plants. So in the seed plants, we have non-flowering plants and flowering plants. Seed plants are the most dominant type of plant on Earth, but most of them happen to bear flowers. I'm wondering if so I said here, right on the top, significant evolutionary milestone for plants, right? So seeds are a big deal for plants. And so how, why, why, if every plant on Earth, if most of the plants on Earth make seeds, there must be a reason. If most plants on Earth make seeds, there must be a reason. And the reason is because plants that bear seeds have an evolutionary advantage. And so now we're going to talk about, well, like what? Well, here is some of those advantages. So seeds are really great at surviving. Seeds, so they really help plants spread to make more plants, much better than the plants we've looked at so far. Part of the reason they do that is because inside of a seed, so part of the reason they do that is, number one, in a seed, the embryo, so the zygote and then the embryo, is inside of a seed. So that alone provides additional protection for the embryo, right? Because it's inside of a seed. Number two, inside of that seed, next to that embryo, is a food source. So it's almost like the plant like packed a little lunch for the baby, right? Like inside the seed is the baby and then food source as well for the embryo. So that really is helpful. That's much better than what we've had before. So the seed plants have this advantage over plants that just shed the spores, right? If you're shedding spores, the spores grow into a gametophyte plant. And then the gametophyte plant has on it egg and sperm that are going to fuse. And then your 2N zygote is going to grow out of that gametophyte plant, right? That 2N zygote and later embryo is not protected. It doesn't have a food source. So in the seed plants now it is protected and has a food source, and that's much better. Yes, so we saw a schematic like this before, which if I seem to recall, I kind of messed it up when I did it before. So where should we start? I guess we'll start here. You see here is mosses. Here, maybe I should switch colors. Let's go red. Right, so mosses, which are, we've talked about way long ago in this class, right? They're well over 400 million years ago is when they showed up. And then those first vascular plants, those early vascular plants, right? They showed up a little later, right? Around 410 million years ago. And then you get to, here, let me get out of this so I can see this, because this thing is blocking my vision. Here we go. Are the first seed plants. So here you go. First seed plants appear around 360, 380 million years ago. And some examples that you could see here are ginkgos, conifers, cycads, which are gymnosperms, which we'll talk about in another class. And then later the flowering plants. Flowering plants showed up much later, about only 130 million years ago, right? But notice when the flowering plants showed up, right? Well, so this is showing you here. First, let's talk about the non-flowering ones, right? So these are showing up around 360 million years ago. And then other ones like the ginkgo, the cycads, they're showing up a little later, right? During the Triassic and the Permian. But notice when the flowering plants show up, right? Which is during the Jurassic mostly. And so maybe when you hear the word Jurassic, maybe you think of like dinosaurs, right? So all these plants here, you know, this would have been things that the dinosaurs eat and later flowering plants. Yes, so when you're looking at these slides later, notice I'm just telling you around when the first seed plants showed up. So this is similar to what we saw when plants made the leap from a life in the ocean to a life on land. Because when that happened, right? When plants went from ocean to land, we saw things like these first amphibians showing up, right? Maybe even some insects. But then later, when plants start to make seeds, which is in that Permian, Triassic, and Jurassic, look at the explosion of animal diversity, right? Tons of animal diversity, because now the animals have a lot more resources to eat, in that they're seed plants. And the fact that there was so much animal diversity is going to allow the seed plants to also diversify because of the pressure of animals eating them. And that's why there's so many flowering plants. Yeah, cool. Right, that's what I just said. So first seed plants show up, and then look at the animal diversification that occurs around that time in the fossil record. All right. So you might have remembered when we first started to talk about heterosperous versus homosperous, one thing I told you guys is the reason why heterosperous is so significant in the evolution of plants is because it ultimately leads to the seed in plants. So here is a nice schematic that kind of organizes where we've come from so far in this class and whether or not the sporophyte plant is homosperous and heterosperous, right? So let's change colors. So with the non-vascular plants, all of them were homosperous, right? All of them were homosperous. When we got to the lycopods, right? Lycopodium was homosperous. And then when we talked about the horsetails and the ferns, most of those were homosperous. But in the lycopods, we met heterosperous for the first time, right? In Selaginella. And there were these aquatic ferns that we talked about that were also heterosperous. So that's where we've come from so far. And you may have noticed a trend here, right? If you go from top, go from up here downwards, you see a trend or a shift from homosporous to heterosporous. And so when we get to the seed plants, both types of seed plants are heterosporous, because that's what I was trying to tell you before, is like you can't have seed plants unless you have sporophytes that are heterosporous. Yes. Okay. So these are some terms that we've already come across in this course, right? Heterosporous, megaspore, microspore. So every seed plant that we talk about for the rest of this class is heterosporous, which means it has two types of spores. It has megaspores and microspores. The megaspores are going to give rise to or hold the female gametophyte. And the microspores are going to give rise to the male gametophyte plant. Yes, and so seed plants are what we call an extreme form of heterospory. And the reason why we call it that is because now we have this new structure, which is the ovule, And it's going to be the ovule which ultimately turns into the seed. So don't worry. We're going to talk about this a lot in this class and subsequent classes. So today, just getting introduced to the seed plants. Don't be too, most of this will make more sense as we talk about it. All right. So what is an ovule? Well, you may have heard the word ovule before, because animals also have ovules. But in plants, an ovule is a structure that contains the female gametophyte plant with an egg cell. And that gametophyte plant inside of the ovule is better protected because there's layers surrounding it, which we'll see on a diagram. After the ovule, right, so an ovule is a structure that contains a female gametophyte plant and an egg. A seed is a structure that contains the zygote, which turns into the embryo. And in other words, let's pick orange, I guess, right? Inside the ovule, you have a female gametophyte with an egg cell. Once that egg cell inside of the ovule is fertilized by a sperm, the result is a 2N zygote, and now we have a seed. So ovule with an egg gets fertilized, and boom, now you have a seed with an embryo inside. This will make more sense, I think, with pictures and not just words. So here we'll learn a little bit about the parts of an ovule first. So here, remember I said an ovule is a structure that has inside of it a female gametophyte and an egg. Let's see, what do I want you to know from here? Okay. I guess I should back up for 1 second. So I should say when the ovule is mature, and finished, like it's done being developed, it will have inside of it female gametophyte with an egg. But just like anything else that has to develop, doesn't just start, like it's not like there's no ovule and then boom, an ovule with a female gametophyte and an egg in it, right? It has to kind of develop. And keep in mind, right, if you're dealing with female gametophyte plant, we know that grew from a spore, right? And if we go back to this previous here, right? if you have a female gametophyte plant, you know that grew from a megaspore. You can't have a female gametophyte plant unless you have a megaspore first. Like I said, this will make more sense as we go forward. This might seem a little bit much today, but don't worry. Don't worry. We're going to go over this a lot. Okay, so here's your ovule that is developing, right? This whole picture is showing you an ovule that's developing. And inside of this ovule is a megaspore. Okay? And that megaspore is going to be what grows into the female gametophyte plant. And as we already know, when you have a megaspore, it's inside of a megasporangium, right? So this whole thing here that I'm doing a horrible job drawing on is a megasporangium. But notice now that the megasporangium is inside of something, which I'm trying to draw here in blue. The megasporangium is not just out on a leaf anymore. Now the megasporangium is inside of something, which is called the integument. So the megasporangium is better protected, which makes the megaspore better protected. And ultimately, when this megaspore grows into a female gametophyte, she's going to be better protected too, because she's inside of this new structure called an integument. So these are some basic parts of the ovule. Don't worry about micropile right now. Don't worry about that right now. So yeah, this slide is telling you Megasporangium, which is here, can also be called nucellus. You'll see that term probably in lab once, that'll be after spring break. Nucellus. And what is the megasporangium? It's the inner part of the ovule, and it's what holds that megaspore. That's what the megasporangium is here in the seed plants. Functional megaspore. So megaspore, we're used to megaspore, but we're maybe not used to this idea of functional megaspore. So just for now, know that this functional megaspore is going to be what it's located inside of that megasporangium, and it's going to grow into the female gametophyte plant. It's going to grow into the female gametophyte plant. And then I think I have integument. Yep. So some seed plants have one integument. Some seed plants have multiple integuments, like so many of them, which is even better protection. But it's basically the outside of the ovule, and it surrounds the megasporangium. After the female gametophyte plant grows from the megaspore and gets fertilized, eventually that integument is going to turn into what's called the seed coat. But this, don't worry about that too much today. You're just kind of getting introduced to these new terms. Oh, and I guess I do tell you what micropile is. So micropile, you might notice, here's the integument, right? This is the integument. And there's a little hole. There's a little hole. So the megasporangium that's inside is a little bit exposed because there's this little hole. And what that little hole is that's where the sperm can enter to be able to meet the egg once there's a female gametophyte plant inside. Okay. Oh, yes. Oh, yes. So this is another big shift in evolutionary things. So think back to the other plants that we dealt with that were heterosporous, right? I think the easiest one to think back to is probably Selaginella. So think back to Selaginella and remember what the strobilus looked like, right? You had Megasporangium on some of the leaves, you had Microsporangium on some of the leaves, but ultimately the megaspore and the microspores got released into the air, right? In the spore plants. Well, now in the seed plants, the megaspores are no longer released. the megaspores stay inside of the megasporangium. And that's a huge shift. It's a huge shift. Oh, you know, I have a logical way of thinking. So look, this is what I was just trying to say with no visuals, right? Remember back in Selaginella? So this is Selaginella. The Megasporangium had inside of it, each one had 4 megaspores inside, and the megaspores would ultimately get released out into the air, But now, and then ultimately those so the megaspores would get released out into the air, and then inside of that megaspore was a female gametophyte plant, right? Who would stick out a little bit, and then a microspore would land on it, and the sperm from the microspore would be able to fertilize the female gametophyte plant that's inside that megaspore, right? But the point is, it got released into air. Now, the megaspore in the seed plants never gets released. It stays inside. And we only have 1 megaspore per megasporangium. So see here, this here, let's actually get rid of this thing. Let's get rid of this. Yeah. So see here, you remember way back when we would have that term, a tetrad of spores, right? Which is a fancy way of saying 4, right? The reason you have a tetrad of spores is because when you have meiosis, the product is ultimately 4 daughter nuclei, right? So 4, tetrad. So inside the megasporangium of, which is here, draw it, of these older plants like Selaginella, each one would have had 4 spores inside of it. You can't see the 4th one. The 4th one's underneath like this, but there would have been 4 inside of the megasporangium. And those 4 would have gotten released back then. Now, in the seed plants, there's only going to be, I don't want these words to get too cumbersome. Inside the Megasporangium now, we're only going to end up with 1 functional megaspore, not 4 anymore, only 1. Only one survives. Which we call the functional megaspore. So remember, inside of, let's take the mega and the micro away, right? Remember, inside of a sporangium, you have cells that are 2N that are going to undergo meiosis to make spores, right? Those spore mother cells. So inside a sporangium, you have sporocytes, aka spore mother cells, right? That are diploid. Those are going to be what undergoes meiosis and cytokinesis, right? Spore mother cell to produce 4 spores. That still happens, but only one of those 4 is going to survive. And that's what we call functional megaspore. I think there's a picture of this coming up. Okay. Question in the chat box. 1 second. Whoops. Guessing the other 3 become food in the seed. That's in the chat box here. That's a good guess, but actually no. The other 3, unfortunately, just go away. They just go away. Other things become food in the seed. Yeah. Other things. But that's a good way of thinking about it, right? Because it's weird to have waste, right? We don't want to have waste here. So what happens to those other 3? Well, in this case, they do just go away. Okay, so why am I showing this to you guys again? Many megaspores function. Okay, so right. This is just to really hit it home. Back when we first learned about heterospore, there were many megaspores per megasporangium, right? 4. Now we're in the seed plants and there's only 1 megaspore per megasporangium, which is what we call the functional megaspore. So what is the functional megaspore? It stays inside of the Megasporangium, right? No longer is released, stays inside. And the functional megaspore is going to be what grows into the female gametophyte plant. It's going to grow into the female gametophyte plant, and the female gametophyte plant is also going to stay inside of here. Okay. Let me see. Let me get out of this. Sorry, it's hard to manage all these boxes here that you guys can't see on your end. I want to see, where do we go from here? Yeah, okay, all right. We should at least go through that. We should at least go through that to get our minds comfortable with some of these terms. So functional megaspore, 1 megaspore now per Megasporangium. Megaspore still going to give rise to the female gametophyte plant, but now female gametophyte plant is also going to never leave the Megasporangium. It's going to stay inside. And the process that occurs that ultimately leads to there being 1 functional megaspore is something that's called Megaspore Genesis. Just remember, when you see these words, break them down. I'm gonna move this thing, break them down. So mega, we know is female something. Sporo refers to spores. And Genesis is like the beginning of something. So the process that leads to the 1 functional megaspore is called Megaspore Genesis, or the beginning of the megaspore. The beginning of the megaspore is one way you could think about that. So here we go on the, let's see, do I give you things? Yeah, good, good. So I don't have to draw it. So let's go through Megaspore Genesis real quick, just to see what happens. So here you go. On the left here, you have an ovule. It's developing. You have a developing ovule with the parts that we looked at on the previous slide. But most importantly, there's a dot in the middle, and that dot in the middle is that sporocyte, right? The spore mother cell that's going to undergo meiosis to produce products, right? So that dot in there is that spore mother cell. This. So that spore mother cell is going to undergo meiosis and produce 4 daughter nuclei, or in this case, 4 daughter cells. But notice that one of the cells gets almost everything via cytokinesis. So notice how this cell at the bottom is so much bigger than the other little ones above. So this is your tetrad. This is a tetrad of spores, the product of meiosis 1 and 2. But look at this unequal cytokinesis, such that the bottom spore is like 5 times as big, maybe even more than the other ones. And that's because it's trying, this unequal cytokinesis allows most of the cellular components to be given to just this one and barely anything else given to the others. And that's why those other 3, those small 3 ones, those are just going to disintegrate and go away. And it's going to be this large one, that large one, which is the functional megaspore. And that functional megaspore, which we can see, like I said here, that bigger one, is going to grow via mitosis and cytokinesis into the female gametophyte plant. So this whole thing now inside is the female gametophyte plant, which stays inside the megaspore as well. So yeah, that's showing you where that functional megaspore is, and it's going to grow into the female gametophyte plant. We already know if you're dealing with a female gametophyte plant, there's going to be an egg on it, right? So eventually sperm is going to fertilize the egg, and the embryo will develop inside of that female gametophyte plant, inside of that megasporangium. So think about how many layers are covering the now embryo. That's kind of what I'm trying to get you guys to be thinking about, is like how better protected, how much protected the egg is, because the female gametophyte plant is way better protected. And then when that egg gets fertilized, that embryo is way better protected as well. This is like protection, 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 which helps survival. Okay. I said that already earlier on. Oh, do I really want to? Oh yeah. Okay, good. All right. We're almost done. We're almost done. Now I know why I'm mentioning this, right? So yeah, so Megaspore never gets released anymore from the Megasporangium. Megaspore stays inside. Megaspore grows into female gametophyte plant inside. Like female gametophyte plant stays inside of that Megasporangium, so better protected. Female gametophyte plant has an egg on it, and when that egg gets fertilized, that's all happening inside of this Megasporangium, and so therefore more protection, more protection. The thing that is causing the protection is this integument, and the integument is, as I showed you at the beginning, it's this layer that surrounds the Megasporangium. Some plants have one integument, some have more than one, but it's this layer that surrounds the Megasporangium, except at the little hole where the sperm can enter. And the reason we think that an integument came about via these ways, right? Via these ways, in order for, and the main way was in terms of evolution, we want the female gametophyte plant and egg to be better protected. We want the embryo to be better protected, but sperm still has to get in there, right? The egg still needs to get fertilized. So there has to be a way for sperm to enter. And that's why we think this integument with micropile evolved. So before I show you that, let's look at this. So here you go. This is a seed. So this female gametophyte plant has already been fertilized, but just notice, because I keep telling you how they're about this better protection stuff, right? So look here, I'm going to do it in red. This whole thing here is the female gametophyte. Her egg already got fertilized. Her egg already got fertilized, such that now there is a 2N embryo inside of the female gametophyte plant. See? Inside, because the egg already got fertilized. But notice that surrounding everything is still this layer. And so before fertilization, that layer, not so good at drawing, was the integument. And now it's going to be the seed coat. Again, these terms we're going to cover many times. I'm just getting you introduced to them today. Yeah. So we know we have an integument and a micropile now. Like we know we have that, but we don't really know the exact order of like how things developed. We just know we have in the fossil record ovules around 360 million years ago. That's when we first start to see them. And here's our best guess about how these ovules came about compared to what we were dealing with before. So if you look here, this is what we think. We think what happened, it's basically like a modified branching system that occurred. So this whole thing here, right? I guess I should do a better job than that. This whole thing here is what is like a primitive ovule, And inside of that ovule would have been a Megasporangium that has a female gametophyte plant. Let's say that that's kind of this yellow thing here, right? Here's another one and another primitive ovule. And what we think happened basically is we're used to the idea of like a megasporangium on a leaf, right? We're used to that idea in, like, for example, Selaginella. But what we think happened is that the megasporangium would have been on a branch or something like that. And then ultimately the stem tissue of surrounding branches kind of would have started to shade it out or to protect it, right? That's what this is trying to show you up here. Oh, I should use a different color. So like look at these long kind of stems that are starting to grow upwards, right? So it's almost like kind of causing some protection. And then it ultimately probably looked more like something like this. See these little kind of stems that are on top of this Megasporangium, which is in yellow. Anyways, this is what we think happened. Don't worry too much about this. Just showing you that you don't just like wake up one day and like whammo, ovules, right? Like there's a process, an evolutionary process that would have occurred in order to get to that point. Oh, and so this idea of like these other stems kind of surrounding a megasporangium is kind of like overtopping when we learned about how megaphils were made, right? Question in the chat box. Yes, so Benjamin says the documentary we watched had a neat animation of this. Yeah, so that's another reason why it's kind of timely to watch that documentary now, because kind of gets you seeing some of these players for the first time, right? That's right. Yeah, you guys watched the ovule first flower one. That's right. That was good. That's good timing. Okay, I think we're almost done here. Let's see. What do I say here? Oh, yeah. Right. So this is just, yeah. If there's things that are just poking out up here to protect, they don't have any egg or megasporangia associated with them. So they're sterile. They're just, but if you have a megasporangium, I guess I should use the same color I used before. You have a megasporangium here, with these kind of the beginnings of an integument surrounding it, that whole structure is fertile. Okay, I think we're almost done. Oh, this is a probably similar to what you saw in the documentary. This is like the evolution of an ovule over time, over millions of years. So let's use yellow. So like yellow here is the megasporangium. So you can't see all of it, but it's in there. Inside here would be a megasporangium. Can't see it because it's inside, etc. And the idea is that initially there would have been this kind of like long stem things kind of serving as some protection, but not like fully protected, just kind of like little fingers. But ultimately those would have kind of closed up, which is what you see here. But sperm still needs to get inside, right? So over time, you'll see like modification where there's a hole kind of toward the top, ultimately leading to the micropile. Okay. Yes. Micropile. So the point of today's lecture is that the ovule is a big deal. It's a big deal. And it really allowed plants to protect female gametophyte with her egg much better. And later, when the ovule becomes a seed, the embryo is way better protected than we've seen previously. So that's a big leap. And it's because of that leap that the majority of plants that we have on Earth today produce seeds. So can't have a seed without an ovule. Can't have a seed without an ovule. So green in this pie chart represents the seed plants. And because the ovule and the seed are such a great thing for plants, we could see that of all the plants that live on Earth, the majority of them shed seeds. So that's the introduction to the amazing ovule. The next classes, we're going to talk about some of these actual seed plants, right? The gymnosperms and the angiosperms. The majority of plants on Earth make seeds, and the majority of those plants also make flowers. And the flowering plants are called angiosperms. So if we looked at that, if we looked at just this green part of this pie chart, which is all the seed plants, and looked at that on a new pie chart, like here, you would see, look, over 360,000 are flowering plants, and just about 11,000 are the gymnosperms. But the gymnosperms, even though they don't make up that many species, still super important. So what do these words mean, angiosperm and gymnosperm? You may have come across this in a previous class, and maybe this was even mentioned in the documentary, could have been. Here, let's use black. Let me move this out of the way. Move this out of the way. Gymnosperm. Gymno means naked, and sperma means seed. So gymnosperms means naked seed. What does that mean? What does that mean, naked seed? It means that there's no ovary, which we haven't learned about yet today, but it means that the ovules of gymnosperms are just exposed. The ovules are just exposed. They're just if you look at a gymnosperm, you can see the ovules with your naked eye. So on the gymnosperms, the ovules and later the seeds, right? Because an ovule will turn into a seed, are naked, which means they are visible. They are visible with the naked eye. They are exposed to the environment. Whereas in the in the angiosperms, angion means a vessel and sperma still means seed. So in the angiosperms, the ovules are inside of a structure called an ovary. And later the seeds will be inside of a structure as well. So in the angiosperms, the ovules are not exposed to the environment. You cannot see the ovules with your naked eye, right? Because they're inside of a structure. We will say the words gymnosperms and angiosperms a lot of times going forward. So this is just getting used to them today. Here's what I mean by inside of something, right? So on the left here, you have the gymnosperm. On the right here, you have an angiosperm. Here are some of the things we just learned, right? We just learned integument, right? Integument. Maybe I'll change colors. It's there's a lot going on this slide. Let's use, I guess, red, right? We just learned what an integument is, right? Here's integument. So the integument in this slide is shown in blue. And notice in the gymnosperm, the integument is not surrounded by anything, right? The integument is naked, just out there. So the whole ovule is just naked. Whereas in the angiosperm, the ovule, right, is inside of a structure, which is called an ovary. And we will learn about that a lot going forward. But just so you get comfortable with these terms, gymnosperm versus angiosperm. Gymnosperm, naked seed, angiosperm, clothed or covered seed. Okay, I think that is it. Here is just a preview of some of the players that we're going to see next time. So the first gymnosperms we'll talk about will be the cycads. Following that, we'll talk about ginkgo. And then lastly, we will talk about conifers, which bear cones. And so the next class, we will talk about the cycads. And I think that's it. Yay. Okay. Yeah. So wow, we actually got through it. That's great. Yeah. So next class, we'll talk about cycads. That way you guys will be in good shape for after the break. What should I say about, oh, you guys have a assignment due by Thursday, right? That covers the ferns. So keep that in mind. It's been available since, I don't remember when I made it available, days ago, but it is due by Thursday at 11 a.m. And then I know it seems like a lot of assignments and stuff right now, but just know you have spring break coming up, and you know, I'm just trying to give you guys more opportunities to get points. So if you don't have any questions, you're free to go. I will say if you did score a C or below on the lecture exam, I would like to go over your exam with you. So please email me so we can set up a time to meet during my office hours. But that's on you to set that up. And if you don't have any questions, you're free to go. And otherwise, I'm here to answer any questions somebody might have right now. Have a good day, you guys.
Hi, Professor.
Yeah.
I was trying to see the questions for the exam and it's not letting me. I was gonna say like, are we able to review like the which ones we got right and which ones we got wrong?
Yes, you know what it is? I probably have to go into the exam and turn on feedback. So I'll do that right now.
Gotcha, because I was a
Actually,
little confused at
here,
first.
yeah, you know what, since I have you here, hold on. Let me just pull it up now and see if this changes it, rather than us have to do email back and forth. So let's just make sure,
Gotcha.
since we're here, let me pull it up. Okay, let me go here. And okay, let's do this. Edit. All right. Oops, wait, not yet. 1 second. One second. Here we go. Edit test options. Okay, so now I'm gonna do this. What's today is the 9th. So we'll pick today.
Yeah.
And we'll pick whatever, 3 a.m. doesn't matter. And then I'll put this, boom, 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 and feedback and submit. All right, so I've changed the settings. Might need to like go out of it and try to come back in, but see if you can see things now.
Gotcha. Give me like one.
Yeah, no, take your time.
Yep, now I could do it. Thank you, Professor. Now I can
Great.
review it.
Okay, perfect. Good. Thank you for letting me know that. Yeah.
Of course.
Oh, and another thing I should say is if anybody got, people already left, but if there's any question that you think you got wrong, you're not sure why you got it wrong, you feel free to also reach out to me in case first, maybe I made a mistake or maybe I just need to explain it better. Yeah, so if there's something you got wrong and you're not sure why, feel free to reach out to me. Okay, guys. Thank you so much. Have a good rest of your day.
Take care, Professor.
You too. Any other questions? All right. If you have any other additional questions, feel free to email me. Have a great day.